Spiderweb drugs. Picture yourself leaning over a dark glass enclosure in a pitch-black laboratory, watching a common garden spider spin its web in the silence. You can hear the microscopic rhythmic tapping of 8 needle-sharp legs sticking and pulling against liquid silk pulled straight from its abdomen. The architecture is flawless, a symmetrical masterpiece of engineering designed to capture prey with geometric perfection. Now imagine injecting that tiny creature with a single drop of liquid caffeine. Within minutes, you notice the rhythmic precision completely disintegrate. The silk turns jagged, erratic, and broken. The spider begins darting aimlessly across the frame, throwing tangled strands into chaotic dead ends, until it simply gives up, leaving behind a useless, broken scribble of silk suspended in the dark. You might assume a primitive arachnid brain would react wildly differently to psychoactive substances than a human brain, but in 1995, scientists at NASA's Marshall Space Flight Center decided to put this exact assumption to the test. Researchers exposed house spiders to varying doses of LSD, marijuana, amphetamines, and caffeine, using computer graphics software to map the resulting web geometry down to the precise angle of every thread. The results shattered traditional biological assumptions. While LSD created a surprisingly uniform web, and cannabis caused the spider to lose focus halfway through, caffeine produced the most catastrophic neurological failure of all. NASA's spatial analysis revealed that caffeine completely destroyed the spider's internal geometric coordinate system. The creature could no longer calculate distance, tension, or spatial symmetry, proving that the chemical pathways controlling motor control, focus, and structural planning in arachnid nervous systems share an astonishing, fundamental overlap with the human brain. When you sip your morning cup of coffee, you feel a sharp jolt of alert energy, believing you are optimizing your mind for productivity. But what NASA visually proved is that caffeine does not enhance focus. It temporarily scrambles your nervous system's spatial and structural assembly lines. The agitation, frantic multitasking, and jagged thought loops you experience after high doses of caffeine are the exact same neurological chaos that caused the spider to abandon its web. Your brain is reacting to the exact same chemical toxicity, revealing that your sophisticated human consciousness relies on the very same fragile bioarchitectural rules as a tiny creature spinning silk in the dark. Fox domestication. Imagine walking through a freezing compound in the heart of Siberia, surrounded by rows of wire cages echoing with the sharp, defensive snarls of wild silver foxes. You reach your hand out toward one of the mesh doors, and instead of a ferocious bite, a fox steps forward, drops its ears, and begins wiggling its hindquarters like an eager golden retriever puppy. You feel its warm, wet nose nudge your fingertips, its tail curling softly as it licks your hand. But when you look closely at this creature, something unsettling strikes you. This is not a wild predator anymore. Its fur is covered in unnatural white patches, its ears are floppy, its skull is shorter, and its vocal cords emit a strange, high-pitched whimper that sounds disturbingly domestic. This transformation was not a natural evolutionary shift. It was an accidental disruption of genetic rules engineered by biologist Dmitry Belyaev starting in 1959 at the Siberian Institute of Cytology and Genetics. Belyaev set out to uncover how ancient humans turned wild wolves into loyal dogs, selecting only the top few % of foxes that showed the least aggression toward humans for breeding. He did not select for physical traits, only for behavioral tameness. Yet, within just 10 generations, the fox's physical biology mutated. Belyaev proved that by selecting for low adrenaline and docility, researchers were delaying the development of neural crest cells, the stem cells responsible for both the adrenal glands and facial cartilage during embryonic development. Because the adrenal system was throttled to suppress fear, the physical markers of wildness, erect ears, camouflage coats, and long snouts, literally failed to form.
Every time you look into the floppy ears of your pet dog or feel a warm sensation of comfort around a cute, domesticated animal, you are reacting to a genetic side effect of hormonal stunt work. What you perceive as innocence and cuteness is actually the physiological footprint of a broken adrenaline system and delayed embryonic stem cells. And more unnervingly, evolutionary biologists suspect that humans undergo self-domestication under the exact same biological rules. Your own cooperative nature, softened facial features, and reduced physical aggression are the result of your ancestors favoring low adrenaline, proving that the visual traits you define as civilized humanity are simply the side effects of a throttled survival drive. Planarian worms. Imagine standing under the harsh fluorescent lights of a biomedical lab, holding a tiny, translucent flatworm known as a planarian in a petri dish. You watch it glide across the shallow water, its tiny dark eye spots reacting to the light. You place a microscopic razor blade directly over its body and slice its head completely off. You watch the severed head float away while the headless body twitches on the plastic surface. Now imagine waiting 2 weeks as that headless stump regenerates a brand new brain, complete with fresh eyes and brand new neural connections. When you place this newly crowned worm back into a complex maze it learned weeks before losing its head, it navigates the paths instantly, making 0 mistakes. For over a century, neuroscientists insisted that memories were strictly encoded within the synaptic connections of the brain. But in a groundbreaking 2013 study published by Dr. Michael Levin at Tufts University, scientists proved that memory is not exclusive to the brain. Researchers trained planarian worms to find food in a specific light-textured zone of a petri dish, sliced off their heads, and allowed them to fully regenerate in complete isolation. The regenerated worms retained the exact memories of the original training. Levin's team demonstrated that memory storage extends far beyond synaptic networks, stored instead throughout the somatic tissue via bioelectric gradients and epigenetic chemical signals. The body cells hold a distributed memory map that automatically reinstalls information into the newly grown brain tissue during cellular regeneration. When you look at your own hands or touch your skin, you assume your memories, habits, and trauma live exclusively inside the soft tissue behind your forehead. But the Planarian experiment reveals a terrifying possibility about your own biological architecture. Your body tissue carries its own bioelectric pattern and cellular memory independent of your conscious brain. The visceral muscle memory you feel when typing, the phantom aches and healed injuries, and the instinctual physical reactions you cannot explain might not be coming from your mind at all. Your memories are written into the very fabric of your flesh, proving that your identity is not just held in your thoughts, but encoded throughout every single cell of your body. Glow pigs. Imagine walking into a dark, pitch-black agricultural facility in South China, surrounded by the rhythmic grunts and shuffling hooves of newborn piglets in their pens. The lights flick off completely, immersing you in shadow. Then, an investigator flips on an ultraviolet blacklight. Suddenly, you catch your breath as 2 piglets in the litter begin to glow with an eerie, radiant neon green light. You reach out your hand and touch their warm, soft snout, expecting chemical paint or a topical dye, but the glowing light is coming from deep within their living tissue. Their skin, their hair, their snout, and even their internal organs radiate an intense bioluminescent hue that pulses softly with every breath they take. This eerie neon glow was not created with dyes or radiation. It was the result of a landmark genetic experiment conducted in 2013 by researchers at South China Agricultural University. Scientists extracted the genetic sequence for green fluorescent protein, or GFP, from a deep-sea Aquaria victoria jellyfish and micro-injected the foreign DNA directly into 10 newly fertilized pig embryos. As the embryos divided and developed, their cellular machinery did not reject the marine code. Instead, the pig cells executed the jellyfish instructions seamlessly, synthesizing glowing proteins in every tissue of their bodies. The 2013 study delivered a profound biological detonation, proving that DNA is not a species-locked blueprint, but a universal, open-source programming language shared across every living organism on Earth, capable of compiling code from deep-sea invertebrates into complex mammalian bodies without error. When you look down at your own hands, you view your body as a distinct human organism, forever separated from the rest of the animal kingdom. But the existence of transgenic animals proves that your genetic code is written in the exact same biological syntax as a jellyfish, a tree, or a pig. Your cellular machinery is so fundamentally compatible with foreign life that if synthetic or animal genes were inserted into your genome, your body would read and execute those foreign instructions without hesitation. You are not a locked human species. You are a running program operating on a universal genetic code that can execute any instruction written into the ledger of life. Mouse ear. Imagine looking down at a glass terrarium in a quiet medical laboratory and seeing a small laboratory mouse scurrying across the bedding. At first glance, it looks like a normal hairless mouse, but as it turns, you freeze in total disbelief. Rising dramatically from the center of its back is a fully formed, three-dimensional human ear. You watch the delicate contours of the cartilage move, flexing beneath the mouse's thin, translucent pink skin as the tiny animal scampers around. You reach in and gently touch the ear with your fingertip. It feels firm, warm, and distinctly human, with live blood vessels pulsing beneath the surface, completely integrated into the mouse's living body. This surreal image was not a hoax or a genetic monster, but a landmark tissue engineering experiment conducted in 1997 by Dr. Charles Vacanti and his team at the University of Massachusetts Medical School. To achieve this, researchers constructed a bioabsorbable polymer scaffold shaped in the exact anatomical structure of an adult human ear. They seeded this synthetic frame with bovine cartilage cells and surgically implanted the entire structure beneath the skin of an immunocompromised hairless mouse. As the mouse's host blood vessels penetrated the synthetic matrix, they nourished the cow cartilage cells, which multiplied and secreted collagen. Over time, the synthetic plastic scaffold dissolved into harmless lactic acid, leaving behind a living, self-sustaining human ear structure supported entirely by the host mouse's circulatory system. When you touch your own ears or feel the cartilage in your nose, you assume your anatomical structures are permanent, unique shapes sculpted exclusively by your human development. But the Vacanti mouse proved that your flesh is remarkably moldable. Given the right structural scaffold, living mammalian tissue can be coerced into growing complex human organs on foreign hosts. Your bodily form is not a rigid biological monument. It is a fluid, modular assembly of living cells waiting for a template to tell them what shape to take, proving that the boundary between your human body and synthetic engineering is far more fragile than you ever imagined. Monkey head. Imagine standing inside a dimly lit surgical operating room in 1970, surrounded by the metallic hum of surgical respirators and the cold smell of antiseptic. On the stainless steel table lies a rhesus monkey, its body completely limp, severed at the neck. But right beside it, connected by a web of clear plastic tubes and vascular shunts, sits its severed head, resting upright. You watch in stunned silence as the monkey's eyelids flutter open,
Its eyes track your movement across the room.
It barks defensively, bares its teeth, and attempts to chew a piece of food placed near its mouth, while its completely detached body lies lifeless inches away.
This terrifying, boundary-shattering procedure was executed in 1970 by neurosurgeon Dr. Robert White at Case Western Reserve University. Dr. White succeeded in transplanting the head of a rhesus monkey onto the body of a donor monkey. To keep the brain alive during the critical transition, White cooled the brain tissue to hypothermic temperatures to halt metabolic decay, while using blood vessel shunts to preserve continuous cerebral circulation. When the monkey regained consciousness, it could hear, smell, taste, and react to its environment for 8 full days. Because spinal cord reattachment was impossible at the time, the monkey was paralyzed from the neck down, but its cognitive mind remained fully operational. Dr. White's medical archive provided undeniable empirical proof that your individual identity, consciousness, and perceptual reality reside entirely within the localized neural tissue of your brain, functioning completely independent of the rest of the body. You think of your selfhood as a unified blend of heart, soul, and physical body, believing that your limbs, chest, and internal organs make you who you are. But Dr. White's grotesque success proves a cold biological truth. Your body is simply a disposable life support suit. Everything that defines your personality, your memories, your fears, and your conscious awareness is a captive prisoner inside your skull. If your head were severed and grafted onto an entirely different torso, your mind would awaken in a foreign body, proving that you are not your arms, legs, or chest. You are merely a conscious brain piloting a biological machine. Rat navigation. Imagine watching a completely blind rat standing at the entrance of a pitch-black, complex wooden maze. Its eyes are milky white and non-functional, yet it moves forward without a single moment of hesitation. You watch it dart through twisting corridors, executing sharp left and right turns at full speed, never bumping into a single wall, navigating straight toward a hidden reward, as if it possessed crystal-clear vision. But when you look closely at the top of the rat's head, you notice a tiny microchip fused directly into its skull, flickering with a faint LED light as it translates invisible magnetic fields around the room into direct neural impulses. This startling display of sensory augmentation was created in a 2015 study led by neuroscientist Yuji Ikagaya at the University of Tokyo. Scientists attached a geomagnetic compass sensor to a microchip and wired its output directly into the auditory and somatosensory cortices of sightless rats. When the rat faced geomagnetic north, the chip delivered tiny electrical pulses to the brain's sensory processing centers. Astonishingly, the rats did not experience these pulses as pain or confusion. Within just tens of minutes, their brains rewired themselves to interpret the electrical signals as a native sense of direction. The 2015 Tokyo study proved the extreme adaptability of mammalian neuroplasticity, demonstrating that the brain is a flexible processor capable of instantly integrating entirely new, non-biological senses into its perceptual reality without needing evolutionary adaptation. You go through life assuming your 5 human senses, sight, sound, touch, taste, and smell, are the absolute limits of how reality can be perceived. But the blind rat experiment reveals that your brain is an unlocked sensory platform, waiting for new inputs. If a geomagnetic sensor or infrared camera were wired into your sensory cortex, your brain would adapt within hours, allowing you to feel magnetic fields or see heat signatures as naturally as you hear sound. Your experience of reality is not a fixed window, it is a customizable interface, proving that the world you perceive is merely a fraction of what your brain is capable of experiencing. Slime mold. Imagine looking down at a glass agar plate resting on a laboratory table, covered in a bright yellow, web-like fungal slime. You lean in closely, expecting a motionless mold, but as you watch through a microscope lens, you realize it is pulsing. Every 60 seconds, internal biofluidic waves surge back and forth through microscopic veins, expanding and contracting like a living heartbeat. Now imagine placing oat flakes across the agar surface in pattern points mirroring 30 major cities surrounding Tokyo. You watch as this single-celled, brainless creature stretches across the plate, exploring every path, and then, within 26 hours, retracts all unnecessary branches, leaving behind a pristine, perfectly optimized transportation grid connecting every food source. This mind-bending display of non-neural intelligence was proved in a landmark 2010 study led by Dr. Toshiyuki Nakagaki, published in the journal Science. Nakagaki exposed the single-celled organism Physarum polycephalum to a spatial layout of food sources corresponding to the geography of Tokyo's railway stations. Without a single brain cell, neuron, or central nervous system, the slime mold calculated route efficiency, fault tolerance, and energy expenditure using biofluidic pulse oscillations. When Nakagaki compared the mold's organic network to the actual Tokyo subway system, which took human civil engineers decades of top-tier mathematical calculations and millions of dollars to design, the slime mold's biological algorithm matched human urban engineering with near identical efficiency, redundancy, and structural balance in just over a day. You spend your life assuming that problem-solving, strategic planning, and intelligence require a complex brain with billions of firing neurons. But the slime mold shatters that core belief about consciousness. An organism without a single nerve cell can calculate spatial efficiency, optimize networks, and outthink human civil engineers using nothing more than fluid pressure and cellular feedback loops. Your brain is not the only architecture capable of processing complex decisions. True intelligence is woven into the very mechanics of living matter, proving that the cellular universe can compute reality without ever needing a conscious thought. Jellyfish aging. Imagine peering into a saltwater tank at a tiny, bell-shaped creature no larger than a fingernail, floating weightlessly through the water. Its translucent tentacles drift behind it as it pulses gently under the light. You take a fine needle and inflict a microscopic injury across its bell, or withhold nutrients to simulate starvation. Under normal biological rules, you expect to watch the creature wither, decompose, and die. But instead, as you watch over the next few days, something astonishing occurs. The tentacles retract, the bell shrinks into a dense sphere, drops to the floor of the tank, and transforms back into a primitive larval polyp, effectively hitting a biological rewind button and starting its entire life cycle over from scratch. This radical defiance of mortality belongs to Turritopsis dohrnii, famously known as the immortal jellyfish, discovered in the Mediterranean in the 1990s. When faced with environmental stress, physical trauma, or old age, this creature undergoes a cellular process called transdifferentiation. Instead of cellular senescence and death, specialized adult cells, such as muscle or nerve cells, strip away their functional identities and revert into unspecialized stem cells. These stem cells then reassemble into a juvenile polyp colony, which eventually buds off into genetically identical, youthful jellyfish. Peer-reviewed marine biology data confirms that Turritopsis dohrnii is the only known organism on Earth capable of infinite biological regression, bypassing natural death by recycling its own DNA into a perpetual loop of youth. You live with the terrifying, undeniable reality that your body is a ticking clock, slowly decaying toward an inevitable end as your cells age and lose their repair capabilities. But the immortal jellyfish proves that aging is not an inescapable law of biology. It is simply a genetic setting. Your cells possess the exact same fundamental molecular machinery, but your evolutionary program lacks the transdifferentiation trigger to reset your cellular clock. Your mortality is not an absolute physical rule, but a hard-coded program, proving that nature has already solved the riddle of eternal youth, while your body remains trapped in the linear march of time.
Two-headed dog. Imagine stepping inside a cold Soviet medical laboratory in 1954, holding your breath against the sharp smell of ether and warm blood. On the operating table before you stands a large adult German Shepherd, but as you move closer, your mind recoils in visceral shock. Surgically grafted onto the side of its neck is the upper torso and head of a small puppy. You watch in disbelief as both heads move independently. When a bowl of milk is placed in front of them, the host dog laps it up, while the grafted puppy head simultaneously laps milk, licks its chops, and snaps playfully at the larger dog's ears, even though the puppy has no lower body, stomach, or internal organs of its own. This grotesque surgical marvel was engineered in 1954 by Soviet scientist Dr. Vladimir Demikov, documented in historical medical archives. Demikov achieved this by carefully microsuturing the blood vessels of the puppy's upper body directly into the carotid artery and jugular vein of the adult German Shepherd host. The host dog's heart pumped oxygenated blood through both brains, sustaining life in two independent central nervous systems simultaneously. Demikov created over 20 two-headed dogs during his research. While the animals survived only days due to immune rejection, Demikov's experiments proved that major organs, vascular systems, and complex anatomical structures could be connected between separate living organisms, laying the foundational vascular techniques that made modern human heart, kidney, and lung transplants possible decades later. When you look at another person, you perceive their physical body as an exclusive, impenetrable sanctuary belonging only to their individual life. But Demikov's terrifying work shattered the absolute boundary of biological individuality. Your vascular system is essentially a universal plumbing network, capable of piping blood into foreign heads and foreign organs if connected with surgical precision. You are not an indivisible, closed biological unit. Your body is a vessel whose life support mechanics can be shared, spliced, and grafted, proving that the boundaries dividing one living creature from another are far more artificial than science ever led you to believe. The surgical, genetic, and neurological anomalies we've explored today prove that biological rules are far more fragile than we were taught. But living organisms are only one layer of the matrix. If you want to see how physics, algorithms, and reality itself bend, click the video on your screen right now. Every scientific glitch, paradox, and anomaly discovered in the last 100 years. I'll see you there.